Hey guys, welcome back to another lecture video for Chem 104. In this lecture video, we are going to begin our discussion of our very first macromolecule, which in this case is carbohydrates for the biochemistry portion of this course. And so I'm pretty sure you guys are familiar with the word carbohydrates. It's been, the main focal point, for example, in dieting commercials and cooking or food blogs, and the list continues. And so we typically associate carbohydrates as a source of energy from our diet. However, that is not the only role that carbohydrates play in our body. Carbohydrates can be used in more of like a support role, for example, for structural purposes. It can also be used in recognition of self and non-self, so basically the immune system. Our ABO blood group is primarily composed of specific types of carbohydrates. And so overall, carbohydrates are composed of mainly these three elements: carbon, hydrogen, and oxygen. And so the typical ratio between carbons, hydrogens, and oxygens for these common carbohydrates is going to be Cn, H2n, On. And so what this is basically telling you is that if you guys have, for example, a 3-carbon carbohydrate, the number of hydrogens is just going to be double the amount of carbons. And so here there would have, this specific compound would have 6 hydrogens, and the number of oxygens would be the same as the number of carbons, so this is going to be 3 as well. And so carbohydrates are commonly found in, for example, potatoes, bread, pasta, rice, gummy bears, candy. It is just found all over the place. And so once again, the main purpose, or I'm sorry, not the main purpose, but one of the purposes that carbohydrates play a role in our body is to provide energy in the form of ATP. And so, for instance, if we have glucose, which is a simple carbohydrate, it's a simple sugar, it's going to undergo a sequence of reactions that's labeled as glycolysis in the Krebs cycle. And all of those electrons that's extracted from glucose is eventually going to be passed down to the electron transport chain, and the electron transport chain provides this chemiosmotic potential to basically produce our biological energy, which is in the form of ATP. And so this process to produce ATP is nothing more than a series of oxidation reduction reactions. And hopefully you guys will be able to study this in greater detail in either your biology classes or a future nursing course or biochemistry course. Now, carbohydrates is not the only macromolecule that has the ability to provide energy for our body. Proteins and lipids can also be oxidized, and they can kind of come in different points of the pathway to once again feed into this main cycle to produce biological energy, which is in the form of ATP. Okay. All right. So carbohydrates in general are produced by plants. I mean, it would be really cool for us to be able to produce our own carbohydrates, but that would take the fun out of eating, because all we would have to do is just walk outside and raise our arms up and we're already eating. So there's really no flavor in light. Anyways, so these green plants, they are going to undergo photosynthesis, which is a biochemical process in which carbon dioxide and water molecule is going to be converted into glucose. And so glucose, which is going to be our carbohydrate, is going to be consumed by, for example, animals such as humans. And we do require oxygen to drive the oxidation process, this idea of cellular respiration, to produce carbon dioxide, water, and energy. And so you guys can think of photosynthesis and cellular respiration in terms of this overall chemical equation as opposites of each other, but their actual mechanism is really, really different. So you can't really equate one to the other in terms of processes, but the idea is that respiration is oxidation, overall photosynthesis is reduction. All right. And so carbohydrates, they also go by another name, which is saccharides. And so saccharides come from a Latin word called saccharum, meaning sugar. And so we can go ahead and use the name saccharides to describe different categories, if you will, of carbohydrates. And so if you guys are looking at, if I draw your attention to this flow chart, if you will, carbohydrates is going to be our umbrella topic. And we can go ahead and break down carbohydrates into simple carbohydrates or complex carbohydrates. And so for simple carbohydrates, we can talk about, monosaccharides or disaccharides. For complex carbohydrates, we can talk about polysaccharides. And so this prefix mono or di represents 1 and 2 respectively. So mono is 1, di is 2. Saccharides means sugar. And so if you have a simple monosaccharide, then that means that specific compound such as glucose, fructose, galactose is just going to be composed of one type of sugar molecule. Whereas a disaccharide, even though it's a simple carbohydrate, it's going to be composed of 2 sugar units, if you will. So maltose, lactose, sucrose are examples. For polysaccharides, here we have this idea of starches, fibers, and glycogen. And so polysaccharides is many or numerous sugar units covalently bonded to each other. And so we will go ahead and discuss these just a little bit more in detail in this lecture video, but definitely a lot more in this lecture video series. And so in terms of physical properties of saccharides are typically sweet tasting, but their relative sweetness varies greatly. And so the reason why these saccharides have different levels of sweetness, if you will, it's more or less due to its biochemical or neurological pathway and signaling that would produce these different interpretations of how we perceive how a specific carbohydrate is sweet. And so if we focus on sucrose just for a second, so sucrose is common table sugar. If you take some granules of sugar and you place it directly in your tongue, whatever that sweetness is to you, we're going to attribute that as our reference point, so it's 100. And so if you were to, try, these pure substances of monosaccharides, disaccharides, sugar alcohols, and artificial sweeteners, we get, these different contrasts, these different levels of sweetness. And so, for example, fructose, its relative sweetness is 175. And fructose is typically found in candy, for instance. So if you were to go to the grocery store, go down the candy aisle, look at back of a package of gummy bears, and if you read, high fructose corn syrup, uh, it's referring to specifically this monosaccharide, which has, a relative sweetness of 175. And so maltose, for instance, it's a sugar that is primarily, it's malt sugar, and this malt sugar is primarily found in specific alcoholic beverages such as beer. And so typically we interpret beer as more of like this bitter, bitter taste, if you will. And so its relative sweetness is 33 compared to sucrose, which is 100. And so maltose is a lot less sweet than, for example, sucrose. However, maltose sugar is definitely sweeter than lactose sugar. So lactose is the sugar, the primary sugar that's in milk. Artificial sweeteners such as saccharin, sucralose, notice that the orders of magnitude in their relative sweetness is in the thousands, not in the hundreds. And these non-carbohydrate artificial sweeteners, all they do is they somehow, bind onto our sweet taste receptors, these proteins, and induces a stronger signal, if you will, that we in our brain interpret as, oh wow, it's like super sweet. And so looking at this compound, this artificial sweetener called neotame, everything in blue is actually an amino acid, so it's aspartic acid. And the addition of this neohexyl group allows this compound, this structural compound, to act as an artificial sweetener. And its relative sweetness is 1 million. So sucrose is 100. So we almost have 4 orders of magnitude in difference between the sweetness of sucrose and neotame. And so just very briefly, you guys don't need to memorize this, but this is the organic structure of saccharin. When you guys are looking at saccharin, when you guys are looking at these structures of these non-carbohydrate artificial sweeteners to that of their carbohydrate counterparts, you'll definitely see that there's very little similarity between them. Anyways, one way to synthesize saccharin is to take toluene and let it undergo sulfonation, and then after it's treated with ammonia and after it's oxidized, it's going to produce this chemical compound. And so this chemical compound, saccharin, is typically found in sweet and low. And so I'm going to go ahead and skip these slides. But the main idea is that you have this, sweet receptor, if you will, T1R, T2R. Once these carbohydrates bind onto the cell, it's going to undergo some type of phosphorylation cascade, allowing this depolarization that will produce an action potential, which releases a neurotransmitter. So basically we have the propagation of the original signal, which is the presence of the sugar, the propagation of this neurotransmitter to all these different neurons is eventually going to reach our brain cells, and it's going to somehow our brain cells are going to interpret it as sweet or something like that. And so this image right here is just what this T1R, T2R receptor, one part of the receptor looks like. This, if you guys are looking at the right hand side, this little cavity here is where your sugar is going to come in, if you will. And so once the sugar molecule is inside this cavity, it's going to change this, it's going to change the conformation of that protein, allowing it to essentially close. And by allowing it to close, it's going to change the conformation of the structure at the bottom. And so this opening and this closing of this loop allows these G proteins to basically break off and induce this phosphorylation cascade, allowing the depolarization to occur in that nerve cell, leading to an action potential and eventually releasing these neurotransmitters. And so these artificial sweeteners, all they do is they mimic the structure in such a way where it can bind onto, or it can bind into the active site of this protein that will induce the same signal, if not a stronger signal, if you will, even though a true carbohydrate, is not present. So in other words, we've created an organic compound that will kind of trick our mind thinking that we have a carbohydrate without actually having a carbohydrate. And so overall, carbohydrates come in two flavors. The flavors that I'm referring to are these functional groups that we discussed in chapter 12. And so carbohydrates can have, actually, let me take a step back. Overall, there's 3 functional groups that can be observed in the linear form of the carbohydrates. And so overall, carbohydrates can be categorized by what you guys see here in pink and what I'm circling in blue. And so from chapter 12, we know that this functional group is an aldehyde. And so we have this carbon that's double bonded to an oxygen. And this carbonyl carbon is once again bonded to a hydrogen. And so we can also have a ketone, right? And so what I've circled in blue will represent the ketone that's present in our carbohydrate. So long story short, carbohydrates comes in 2 flavors. It can come in as an aldehyde or a ketone. And we're going to go ahead and look at their chemistry in just a little bit. All right, so we're going to go ahead and talk about the characteristics of monosaccharides. So we've learned that overall we can classify carbohydrates into 3 main groups. There's actually another one. It's called oligosaccharide, and I'll cover that much later. And so monosaccharides, we have this singular sugar unit. And so disaccharide, here we have 2 units. Polysaccharide, we have many units of sugar molecules. And so overall, what we're going to be doing is we're going to be talking about the characteristics and some of the features of monosaccharides. And then we're also going to do the same thing for disaccharides, polysaccharides, and oligosaccharides. So that's basically how this chapter is broken up. And so monosaccharides in general is going to be our simplest carbohydrates just in general. They cannot be split into other forms of sugars. And so if you guys have a monosaccharide and if you treat it with acid or an enzyme, there's really no degradation of that molecule. And so if there is no degradation of that molecule, then we know that a specific substance is a monosaccharide. If we have a disaccharide and if we treat it with acid or some type of enzyme, and it breaks apart into 2 sugar molecules, then we know that the original substance was not a monosaccharide. Okay. And so generally monosaccharides have 3 to 8 carbons, so they're 3 to 8 carbons long. They can be observed as a chain, and they can be observed in a cyclical form. Now, the cyclical form is the most thermodynamic stable conformation, if you will, of sugars just in general. And we'll talk about that in a future lecture video. And so, like what I mentioned earlier, aldehydes and ketones are the two flavors for these monosaccharides. They also, what you guys also see are alcohols, right, or hydroxyl groups. And so, since hydroxyl groups are found extensively in saccharides just in general, we don't really use alcohols to distinguish one type of monosaccharide to another. What we're going to be using is the aldehyde functional group and the ketone functional group to distinguish or classify one specific type of saccharide compared to another. All right, and so the most simplest monosaccharide, simplest aldose, so to speak, is going to be glyceraldehyde, and the simplest ketose is going to be dihydroxyacetone. And so if you guys are looking at this, this image right here at the bottom, here we have a 1, 2, 3 carbon carbohydrate, and here we have a 1, 2, 3 carbon carbohydrate. And so if you guys were to count the number of carbons, hydrogens, and oxygens, then you would see that glyceraldehyde and dihydroxyacetone, one is an aldose and one's a ketose, they're just going to be structural isomers of each other. So if you guys recall from a previous lecture or a previous chapter, constitutional structural isomers have the same chemical formula, but how the elements are arranged, or structurally arranged, are completely different. Okay? And so here we see the idea of structural isomers producing two specifically different organic compounds with different chemistry, different properties, different physical properties. All right, so talking about physical properties for monosaccharides, this can also be applied for disaccharides. Typically, these monosaccharides have a high melting point. And they're also polar compounds. So let's talk about this idea of polarity first. And so if I zoom in this structure over here, notice that we have an extensive amount of hydroxyl groups. So these are alcohols. And so since we have, a plethora of hydroxyl groups, if you guys recall their physical properties, we know that these hydroxyl groups are polar. Therefore, it can interact with water molecules through hydrogen bonds. And so I'm just going to go ahead and draw this here very quickly. So we know that there is a permanent dipole pointing towards oxygen, making this oxygen partially negative, making this hydrogen partially positive. And so if we have, for example, a water molecule, once again, there's going to be a permanent dipole pointing towards oxygen, making this partially negative and this hydrogen partially positive. And so the interaction with the partial positive and partial negative is known as a hydrogen bond between this water molecule and the hydroxyl group in this sugar molecule. And so since there's so many hydroxyl groups, one can imagine that these hydroxyl groups can really heavily interact with these water molecules, allowing it to be very water soluble. Okay, so this is the reason why when you guys take table sugar and you guys were to place it in water and you stir it around, that sugar automatically dissolves if if that solution is still unsaturated. And so because there are so many, hydroxyl groups, there's many points of contacts, if you will, for hydrogen bonds to occur. And so if you guys have a crystal of these molecules, so to speak, the extensive amount of hydrogen bonds between or among these sugar molecules, it's very high. And so if there's a lot of hydrogen bonds, and since these hydrogen bonds are stronger intermolecular forces compared to dipole-dipole and London dispersion force, then it would take a lot of energy for it to kind of separate, for it to, for example, melt. And so this is very important in cooking. I'm not too sure if you guys have done sugar work before in terms of, for example, making caramel or even using sugar as like a decorative piece. If you guys watch the cooking channel, you guys will see this very often. But the melting point is very, very specific for different types of sugars. And so the idea is if you wanted to make, like, for example, a caramel, you would use this candy thermometer and make sure that you solely reach a specific temperature, make sure that temperature is stable. And if you guys have obtained that temperature, then you guys see this nice melting of your sugar, and obviously you guys can add on to it to give it different textures, to give your caramel different texture. But if you guys go past the melting point, then you guys are going to burn your sugars, and then it becomes very bitter. And so, the physical properties of monosaccharides, it's not only relevant in lab, it's also very relevant in the culinary industry. All right, and so what we're going to do now is we're going to learn how to classify our monosaccharides. And so we're going to use two key features, if you will, to classify them. And so the first thing that we're going to do is we are going to classify our monosaccharides through their functional groups. And so if you guys have an aldehyde, what we're going to, so if your carbohydrate has an aldehyde in its functional group, then we're going to go ahead and name that specific compound, or classify that specific compound as an aldose. And so if you guys have a ketone, then it would be a ketose. And so, by the way, the ending, the suffix OSE, is very specific to, for example, to carbohydrates. So for example, glucose, right? So here we see the OSE, fructose, we see that OSE. So the word the suffix OSE, it's supposed to bring up or link up in our heads this idea of a carbohydrate. Okay. And so the second way that we're going to classify our sugars is through the number of carbons. And you guys are going to be using prefixes such as tri, tet, pent, hex, hept. Okay. And so tri means 3, tet means 4, pent is 5, hex is 6, hept is 7, oct, non, etc. So oct 8, non 9, dec 10. And so what we're going to do is we're going to use a combination of the functional groups and the number of carbons to classify our monosaccharide. And so we're going to go ahead and look at a couple of examples. And so the first thing I'm going to do is I'm going to identify my main functional group. And I know that all of these sugars have hydroxyl groups. That's why I'm not highlighting them. I'm just highlighting whether or not it's an aldehyde or a ketone. And so here, this is an aldehyde. Okay. And then I'm going to go ahead and change my highlighter color. So here I have 1, 2, 3. So I have a 3-carbon carbohydrate with an aldehyde functional group. And so I'm going to go ahead and name this as aldo triose. Okay. So aldo is coming from aldehyde. Tri, which is in this case pronounced tree, is going to come from the number of carbon. Then the OSE ending is going to be the suffix that's describing that this is a sugar or saccharide or a carbohydrate. So aldotriose is how we're going to classify this 3-carbon carbohydrate with an aldehyde functional group. And so let's go ahead and go through the next one. Here we have another aldehyde. Oops. And so we have 1, 2, 3, 4 carbons. So since this is a 4-carbon carbohydrate with an aldehyde functional group, it's going to be called aldo. Then 4 carbons, we typically think about that as tetra, but we're going to go ahead and shorten it up to tet, and then we're going to add the suffix -ose. Okay, so this is aldotetrose. Okay, once again, aldo is the aldehyde, tet, or maybe the tet with the R, is going to be the 4-carbon compound, and the OSE is going to be the sugar. All right, so looking at these last 3 examples, here I see an aldehyde, I see an aldehyde, so both of these are going to start with aldo. And so here, oops, I have 1, 2, 3, 4, 5 carbons, so that's going to be aldopentose. 1, 2, 3, 4, 5, 6 carbons, and then that's going to be aldohexose. And so if you guys are looking at the last example here, we have a carbonyl group that is wedged right in between 2 carbons. And so all of this is going to be a ketone. And so I'm probably going to be using the prefix ket or ket. So now I need to determine the number of carbons. I have 1, 2, 3, 4, 5, 6. So I have keto hex because I have 6 carbons. And since this is a carbohydrate, it's going to be ose. So this is going to be a ketohexose. Okay. And so this is just an example of how we can, count the number of carbons, recognize the functional group that is different, the main functional group that's different between these two carbohydrates, and then kind of arrange them in such a way that we create this, this way of naming or classifying these carbohydrates: aldopentose, aldohexose, ketohexose. And so the next two slides, I am just going to show you the different types of sugars. And so please recall that 3 carbons is the minimum amount of carbons required for a carbohydrate, for that carbon to be, for that molecule to be classified as a carbohydrate. And so here in this case, since I have a ketone, this would be an example of a keto triose. Since here I have 4 carbons, 1, 2, 3, 4, and here I see a ketone functional group, I'm going to classify this as a keto tetrose. And so here, since I have 1, 2, 3, 4, 5, so this is a 5 carbon carbohydrate, and it has a ketone functional group. So this is going to be keto pentose. Okay. And last but not least, here if I have a 6 carbon sugar, I have 1, 2, 3, 4, 5, 6 carbon. And once again, here I see a ketone functional group. This is going to be a ketohexose. Okay. And so all of these, it's just the classification of the names. The actual names in itself, there is no specific pattern. If this was a true biochemistry course, this is something that you guys would actually memorize in terms of name and structure. But this is not the full year of biochemistry, so you guys do not need to memorize any of these. This is just a point of reference, but you guys should be able to learn how to classify them. And so this slide right here just represents the different types of aldehydes. So remember that CHO is an abbreviation for our aldehydes. And so we can have an aldotriose, an aldotetrose, an aldopentose, and an aldohexose. Okay? So in each and every single one of that specific classification, there's the names of the actual sugars is very specific. All right, and so that is it for this lecture video. We are going to move on to our next topic in the next lecture video. See you guys later. Bye.